think, yes, I think it's 8:02, and I see that a lot of students are already here. Hi, everyone. Good morning. I'm Stella Christie. I'm a professor at Tsinghua University, and very excited to have Mitya, who has been so kind to again give the lecture. So this is the second installment of the lecture. And as you all know that we have said many times that we ask the lecturers to give their lecture twice to accommodate all of your students' time zone around the world. So, and this is very unusual. So our lecturer has been very kindly agreed to give their time to give their second lecture. I actually listened to Mitya's lecture yesterday, and I'm very still excited to listen to it again. And just to remind the students that you are, Mitya will say it as well, but you are feel free to interrupt if you have any questions. And in fact, I think our lecturer welcomes that so that it doesn't feel very lonely to be up there talking to like a computer. So students are very much encouraged to interrupt and ask questions anytime. Without further ado, I give the floor to Mitya.
Thank you very much. Thanks again for having me speak in this school. So I want to repeat the invitation to please interrupt me and ask questions when you don't understand something. You can do it through the chat, or you can just speak up. All right. So today I want to tell you about an attempt to construct an algorithmic or mathematical theory of a neuronal computation on the so-called mesoscopic level, on the level which is most appropriate to relate to the newly available data sets from connectomics and neuronal population activity imaging. And you will see how that works. But first, I want to discuss a little bit what I mean by the normative approach. So the normative approach implies a systematic derivation of some rule based on optimization. And I would say that optimization approach is rooted in physics. So I think it's appropriate to put up this quote from Richard Feynman, who was one of the leading physicists of the last century. And he said, in general, we look for a new law by the following process. First, we guess it, then we compute the consequences of the guess to see what would be implied. If this law that we guessed is right, then we compare the result of the computation to nature with experiment or experience, compare it directly with observation to see if it works. If it disagrees with experiment, it is wrong. That is all there is to it. So sounds almost trivial, but this kind of simple prescription, I think, was responsible for the tremendous success of physics in the last century. So I want to take it seriously and apply it to neuroscience. But let's first unpack a little bit what Feynman is saying. So first he said, well, we guessed the law. So in physics, what that usually means, it means learning some kind of objective function or energy function. In physics, it's usually free energy or Lagrangian or something like that. And then starting with this optimization problem in terms of this energy function, one performs an optimization on it, and the solution to this optimization results in the equations of motion in physics or some kind of dynamics in general. And that's the kind of rules that can be actually compared with experiment. Okay, so optimization provides this bridge between the guess, the energy function, and the predictions, which usually are formulated in terms of dynamics. And now in physics, the energy function is guessed usually based on some prior experience, maybe some on symmetries, on some symmetry considerations. But that's not the case in neuroscience or biology in general, because biological systems are there to perform some function. Otherwise, they wouldn't be there. And so it's very important to realize that when you write down an energy function, it's supposed to optimize your performance or the organism's performance on some task. Of course, which task we don't know, that's what we have to guess. And that's where creativity comes in. And so today, I will start with the traditional guess that has been made about 40 years ago in neuroscience, which I would call the reconstruction decoding approach. This approach had many successes and predictions that have been verified experimentally. And we will discuss all that, and we can cover it all using the single neuron case. But we will see that when we try to expand the reconstruction approach to the multiple neuron case, we run into some problems. And in order to preserve all the successes of the reconstruction approach, but to overcome this problem, we developed a new similarity matching approach, which will be the subject of the second lecture. So today we'll just focus on the review of the traditional reconstruction approach. Okay, any questions so far? Okay, let's go on then. So to introduce the reconstruction approach, the best way to do that, I think, is to start with the assumption that because we are aware of the world and our surroundings, there must be some kind of neural representation of the world. And that representation is probably somehow stored in the activities of the neurons. To make this a little bit more specific, let's consider visual system. And that's when you have a sensory stimulus, like shown here. And that sensory stimulus is represented in the visual cortex, in the activities of the neurons in V1, which we know from the time of Hubel and Wiesel respond best to edges of varying orientation and position. So when the stimulus is presented to the brain, the activity of the neurons changes to represent or reconstruct that stimulus. To make this mathematical, we write down the light intensity in each pixel as components of the vector x. So we take the light intensity in this patch, this red square, and we write down the intensity of the first pixel as x1, second as x2, and so on. And that is this bold-face x, which is a vector of light intensity, the stimulus. And we are postulating, right? It's a guess. We're postulating that that x is represented by the activities of the neurons y's through their feature vectors. A feature vector of each neuron will be the stimulus that that neuron responds to the most. So for example, here you can see the feature vector of one of the neurons, which is this Gabor patch, as you know, feature vector of another neuron, and so on. And so these are all different Gabor patches of different orientation. And you can see that they form some kind of a basis. These are not orthogonal functions. So strictly mathematically speaking, it's not a basis, it's a dictionary. Nevertheless, you can expand any image as a linear combination of this kind of Gabor patches. And the coefficients in these expansions are this YKs, which are the activities of the neurons in the visual cortex. Okay, so we assume that we can linearly reconstruct or decode the visual stimulus by weighting the feature vectors WK by the activities of individual neurons. Now, by activities of neurons, I will mean a real number, which could be thought of as a firing rate, for example, for spiky neurons, or graded potential, membrane potential for graded potential neurons, we're really not going, in this talk, we're not going into the details of spike timing. So we just represent the activity of neurons by a real number. Also, I should make a point that, just because we would like to view the activity of neurons as a representation of the world and reconstruct the world linearly from that activity, doesn't mean that we think that that's what the brain is actually doing. In the extreme sense, this kind of view is called the homunculus fallacy, right? When homunculus is this little humanoid, okay, that sits inside your brain and watches the TV on which, the neurons just like light up like this, like shown here, okay? And then it has to figure out what's going on. Of course, that's not what happens, okay? In reality, the brain doesn't need to reconstruct the world from the activity of neurons in V1. The only thing that the brain needs to do is to generate behavior. And that behavior has to be appropriate to the particular sensory input that it gets, okay? But the reason people came up with the reconstruction objective is to take a shortcut, because historically it has been easier to understand the sensory side of the brain than the motor control. And so to make predictions about the sensory side, they came up with this reconstruction objective. The main consideration here is to make sure that information in the images is not lost in the neuronal activity in V1, okay? So that's the basic idea of the reconstruction approach. Any questions?
I actually would like to ask a question here, if you don't mind, Mitya. So what are the constraints, the factor of the neurons factor itself? So my question is, because just now you said that, I mean, obviously the brain, the reconstruction is sort of a shortcut, so that the sensory stimulus are not lost. But we know that in actual vision and perception, a lot of things are lost, and those losses are, so to speak, have beneficial advantages, right? So we're not a video camera that that faithfully reconstruct everything from the from the environment. So what are the constraints like here already? I mean, are there already constraints within the linear decoding approach that you're using?
Yeah, so that is a great question, and your criticism is absolutely correct. And in fact, the decoder approach may work well at the very early front end of vision.
Okay.
But you cannot keep applying it. Like if you want to build a deep network, you cannot keep applying this approach, because that's completely not what you want to do. You don't want to keep reconstructing the world deeper and deeper into the brain. You want to come up with some kind of latent variables that will allow you to generate the behavior. So that's just the limitation of the reconstruction approach.
Okay.
And, we will try to overcome it a little bit, but I think that that will require serious rethinking in the future. Yes.
Thanks.
What other, okay, so there is a question in the chat. What are the reasons that we are only using linear decoding? That's a good question. And the reason is just simplicity, just because we want to make progress. So we are assuming that we can do that. The hint that this may work is that the number of neurons in the visual cortex is actually much greater, maybe 2 or 3 orders of magnitude greater than number of channels of information that are coming from the retina in the optic nerve. And so this is a higher dimensional representation in the visual cortex. And it is often possible to linearly reconstruct things from the higher dimensional presentation, even if the forward transformation, the encoding, is nonlinear. Okay, so you can have nonlinear encoding, but linear decoding if you have the expansion of dimensionality. So that's a good question, but that's what we'll stick for with for simplicity for now. In second lecture, we will do something nonlinear. Okay. All right. So so the plan is going to be the following. What we're going to do now, we are going to take this reconstruction approach, linear decoding assumption, and turn it into an objective function. And then we will use this objective function to derive neuronal dynamics and synaptic learning rules that will be able to reconstruct the world just from the data. So we will assume that all the learning happens in an activity-dependent fashion as the brain is developing, okay? We will assume that the genes just lay down the number of neurons and the general architecture, but all the synaptic connections in the strengths of synaptic connections are determined by the learning. And so we will try to derive the learning rules that will learn the kind of receptive fields that you're seeing here, the edge detectors, that that will learn them just from the natural stimuli. Just if you show a series of natural image photographs to the algorithm, it will learn those images. So that will be our goal. So it's natural to write down an objective function in this form because we said, well, we'll assume x is equal to wy, but what if it's not equal? Then we will penalize the deviation by writing an objective as a reconstruction error squared, sum over all time step t. Okay, so the index t is a time step because we keep showing image after image, and it's both the time step and the index of the image. And at each time, and we're considering just a case of a single neuron here, okay? That neuron's activity in response to that stimulus is yt, and the w vector will correspond to the weights of synapses impinging on that neuron. So that's our goal, but we want to learn those w's and to compute y's through optimization. So before we do that, I want to ask you a question. So you have this objective function for some data set X, and you want to find optimal W and Y. Does anyone know what statistical algorithm will solve this problem? What kind of solution, W or Y, would it have in the optimum?
What algorithm would we use?
Yeah.
No sé, I don't know, something like gradient descent, maybe?
Okay, very good. That's one option. And that often works. But in this particular case, we can actually guess, we can run gradient descent, it's perfectly fine, but we can also guess what the solution will be once the gradient descent converges.
Something perpendicular, maybe. I don't know, something more geometric. I don't know.
Yeah, you're thinking the right direction. Okay, so this is actually principal component analysis. The reason it may not be so obvious because usually principal component analysis is introduced differently. If you have a data set, so in this case, a two-dimensional data set when you have two components, X1 and X2, and here is one data point and there's the whole cloud, the whole data set, then usually principal component analysis is introduced as to find a direction of maximum variance, W, okay? But actually it turns out that the formulation that I've given you in terms of the reconstruction error squared and maximum variance are mathematically identical. And you can do this as a homework problem to show that they are identical. But for now, I will just illustrate graphically what those reconstruction errors are. For each data point xt, we project onto W and that allows us to compute YT. And the difference between the projections and the data point is the reconstruction error shown in blue. And the sum of the squares of the norms of those reconstruction errors is what is the objective function computes, okay? And so that means that the W will have to kind of find the optimal direction so it fits all the data points as well as possible. So find some direction like this. And that exactly is the direction of maximum variance as well. I see that in the chat, there is a question. Is the W does W depend on T? If not, does this assume static coding? Okay, this is an excellent question because it just precedes what I was going to say. I'll answer it in a second. But before that, I want to say that if the whole data set is already given to you, you've seen all the X's, okay? Then to compute W, what you do is to compute the principal eigenvector of the input covariance, right? So you compute the input covariance matrix by adding the outer products, and then you perform the eigendecomposition, and then the eigenvector corresponding to the biggest eigenvalue is W, okay? So that's the solution of this problem in the so-called offline setting, like a statistician would do when all the data is on the computer in memory. But as asked in this question, the neuron rather, does not operate in this offline setting because the neuron doesn't have access to the whole data set at once ever, right? Because the data are this activity vectors, one vector at a time, that is streamed to the neuron by sensory organs. In terms of this activity vector, the components of which are X1, which is like a firing neuron of, say, receptor 1, X2, firing rate of receptor 2, and so on. And they all impinge on this neuron. And so at any given time, the neuron has access just to one vector of activity, XT. And it cannot wait to see the whole data set go by, because by then the animal will be dead. Game over, right? So Y has to be computed on the fly while the X is being presented, okay? And then W is this vector. Of course, there is one value that is optimal for the whole data set. But as correctly pointed out in the question, because the data set has not been seen yet, the W has to be adjusted with every presentation XT a little bit. So each point XT should pull W towards itself, okay? So yes, in the online setting, the W keeps constantly changing. Y has to be computed on the fly approximately with the existing W, and no significant fraction of the data can be stored by the neuron, because the neuron is not a computer. It doesn't have random access memory, so it cannot store any significant data. It can only store information in synaptic weights, which is just like a vector of the same dimension as the data. Does that help with the question in the chat? All right. Any other questions? Okay. So this problem, the online PCA that cannot be solved by this eigendecomposition is shown by animation here, okay, when the data comes in and the W, the Y gets computed on the fly and W gets adjusted. You can see how it works, okay? So this is the so-called online PCA, and it is, of course, harder to solve than the offline PCA. But such algorithm was actually proposed by Oya in 1982, and he actually proposed it in the context of a simplified neuron model. He suggested that a neuron could actually be viewed, obviously an oversimplified neuron can be viewed as performing this kind of computation. How does that algorithm work? Well, as you can see in this optimization, there are actually two optimizations folded in. The optimization with respect to W and with respect to Y. And what Oya's algorithm suggests is that when each data point arrives, the algorithm performs in two phases. The first phase is computing optimal Y while using a fixed W from the previous time step. And in the second phase, we will adjust the W. Okay, so let's do the first phase first. It's actually pretty simple algebra, but I don't expect you to follow it on Zoom. I just want to show you to illustrate that it's basically very simple. You expand the square, or keep only the terms that depend on Y, take W, norm W squared outside of the brackets, and then complete the square. And then you get that YT is actually an inner product of the W with X normalized by W. So this is now starting to look like the model of a neuron that you're used to, where the output is the weighted input, where the weights are encoded in synaptic weights. And physiologically, of course, you know that a neuron can implement that very easily, because every time there is a spike that comes into one axon, it hits a synapse, and then it is turned into postsynaptic potential, right? I'll draw it here, postsynaptic potential. And the magnitude of this postsynaptic potential is associated with this, with the value W, okay? So these are now currents in the dendrites, and they can be summed by Kirchhoff law in the dendrites and the body of that neuron and get converted into its output Y, okay? So we get that a result very familiar and something that can be natively implemented by a neuron, but we got it through the normative approach by optimizing an objective function. And that's good news, because now we know what the result of the computation is. Okay, so now let's do the phase two, where we optimize not just the error, reconstruction error times step t, but the sum of all the reconstruction errors squared seen up to now with respect to W, while keeping YT what it is. And again, this is pretty simple algebra. The important thing to realize is that in the end, W is given by the sum of previously seen data points weighted by the corresponding outputs of the neuron YT, and then normalized by the cumulative activity. Now, I made this big deal about neuron working in the online setting without having to remember all the data that it has seen so far. But in the solution here are all the sums. So how are we going to compute W? Well, it turns out that again, through simple algebra, we can turn those sums into a recursion relation for W, which updates W to the amount proportional to the reconstruction error for the last representation, scaled by Y. This is very easy to understand intuitively, because each data point, when presented, pulls the vector W towards itself. And that's this reconstruction error term that reflects that pull. But the magnitude of the pull is given by the YT, which is the response of the neuron. Why is it there? Well, because if the stimulus was completely orthogonal to W, which is represented by the neuron, then this neuron has no business represented that stimulus, and the W shouldn't be adapted to it. some other neuron will take care of it. Only if the data point is co-aligned with W, then you should take this into account and move W in the direction of this data. Finally, there is this last term, which is the learning rate. In this case, it's the sum of the previous neuronal activity squared. And again, you don't really need to remember all previous activities to compute this. All you have to do is a running sum, which you update by adding the activity at a given point, which could be done through some kind of calcium concentration, which goes up when neuron spikes a lot. And then calcium could modulate gene expression, say through the CAM kinase pathway or something like that. Anyway, a cell is a rather complicated beast. And so to store one scalar variable per cell is not really that hard. Okay, so this kind of expression, this recursion, is called a learning rule. And in this particular case, it is a Hebbian learning rule. And what is meant by that is that W is given by the covariance of the postsynaptic and presynaptic neuronal activity. That's what makes it Hebbian. Any questions?
We called X the inputs, right? Or,
yeah.
I don't remember. Okay. Do you actually apply this model to measures that fine of dendrites? I don't know. I don't know if I'm lost or.
You mean to test this?
Yeah. What data do you need to test this?
Again, you're preempting my next, the slide after the next. So I'll answer this in the next couple of slides. Any other questions? Yeah, and while we're waiting, there are other questions. I should just quickly answer one thing: that Hebbian learning rules have been proposed based on experimental observations, and so the general idea that neurons that fire together, wire together, has been embedded in neuroscience for a long time. And so there is a whole family of Hebbian rules. This one particular Hebbian rules, due to Oya, is the one we use. But in general, there is not enough data to differentiate among which of them is actually implemented. But the fact that Hebbian rules are at work in the brain, I would say are accepted pretty widely in neuroscience.
Mitya, I still have a question in terms of
yes.
A specific form here, xt minus w times yt, right?
Yeah.
How do you think this term is specifically implemented by the neuron?
That's actually a very good question, because the bold-faced letters are vectors, and so
right.
Actually for each individual neuron we only need xt1 minus w1 yt, and I think that they're not actually. I suspect that this difference is not explicitly represented. What is represented in the brain, in the neuron, is the product y times xt, okay? Because you get y through dendritic backpropagation, you get xt through spikes, and then you subtract w times yt squared.
Yeah.
Okay, so I think that these terms are represented separately by different molecules in the synapse. if that's what the synapse is doing. Okay. If there are no questions, let me summarize what we've done so far. So we're following up on what Feynman said, and we're developing this normative approach. The foundation of this is to come up with an optimization problem that relates to biological function. In this case, we took a very simplified version, which is this reconstruction error squared. We first formulated this optimization problem offline because offline, we know what the exact solution of optimization is. So we know what task is being performed, right? That's PCA. That's the top eigenvector of the input covariance. But neurons cannot operate in the offline setting. They have to operate in the online setting. And to perform optimization in the online setting, we alternated the optimizations with respect to Y and with respect to W, which are done in each time step after the new XT arrives. And respectively, they give rise to the dynamics of neuronal activity, which is this weighted summation of the inputs, and the synaptic weight update, this Euler rule that we just discussed. Together, these two steps are could be natively implemented by a biological neuron, which is naturally built to perform weighted summation and also capable of updating the synaptic weight. And this last point is worth dwelling on a little bit because I want to emphasize something that will be very important in this talk, which is that this update, okay, involves only the quantities that are physically available to the synapse. So as I was answering Trent's question, the synaptic update has to know the output activity, the postsynaptic neuron activity Y, which is known because there is this dendritic backpropagation that sends the spike back into the dendrite so that synapses know about it. Each component of W knows the corresponding component of X because these are the spikes that arrive presynaptically, so the synapse has access to it. And the rest is the synaptic own weight, obviously it knows it, and again, the postsynaptic activity, and then the cumulative activity that we decided we can store in terms of some sort of gene expression profile or gene product or something like that. But it's important. Yeah, please.
Can I just ask you, you're talking about the inputs coming into the dendrite specifically. Is there any difference for inputs arriving at different locations?
So that's a very good question. There is a lot of evidence that location is important, but for the purposes of this talk, we will just ignore it and we will assume that a neuron is a single electrotonic compartment. If I have time in the second lecture, I have a piece, I don't know if I will be able to get to it, and we will consider what happens in a two-compartment neuron. But so far, we just ignore this issue.
Thank you.
Okay. Okay, so, but just to emphasize this, that to update a synaptic weight, the synapse needs to know activity of just the two neurons it connects. And so these are the quantities that the synapse has. It doesn't need to know the activity of any other neurons in the system. Why am I so trying to be so adamant about this? Because this is the only kind of learning rule that a synapse can implement. And there are many machine learning algorithms, such as, for example, well-known backpropagation algorithm used in deep learning that use non-local learning rules that update synaptic weights based on the activities of neurons other than the synapse that connects it, okay? And so these kind of algorithms are not biologically plausible. There is no way that this specific algorithm could be implemented in the brain. So that's why there is a cottage industry now of people trying to come up with biological deep learning. But so far, I'm not aware of equally powerful algorithm. And so what we try to do is to develop an alternative to this, starting in this case from unsupervised setting. Okay? Oh, there is a question in the chat. Can online PCA inverse optimize? I'm not sure I understood what you're saying. Are you asking? Are you asking whether synaptic weights can be computed before the activity? Yeah, so you could write it this way. Yeah, it doesn't matter. But you would have then to take the y from the previous step, take the y from the previous step, and then use that y with the previous x. So you look, I wrote it down as two alternating steps, but in biology, those two steps are probably operating at the same time. And so it just, biology probably takes advantage of the difference in time scales, and they are carried out simultaneously. I don't think there is any depth to me saying that first we find optimal y and then w. I think it could be done concurrently. Okay, so with the summary of what we've done, as I already mentioned before, in response to one of the questions, it's time now to compare our predictions with experiment. Just like Feynman said, okay, this is our test. And the first thing that we are going to test is the prediction that the synaptic weight vector is the top eigenvector of the input covariance matrix. Okay, so this may seem like a simple prediction, but it's very difficult to find data to test something like this, because you need to know all the synaptic weights impinging on a given neuron and the synaptic and the activity pattern for all the neurons upstream of this one over a decent period of time to compute the covariance matrix. But we were fortunate enough to identify one neural circuit where both data sets exist. And this is a peripheral olfactory system of a fly larva. It's a very small circuit which has 21 olfactory receptor neurons. Each of those neurons contains a different olfactory receptor set, which has a sensitivity to different odors. And all of those 21 orans, before sending their signal downstream to the central brain, the mushroom body, projects onto interneuron I. And it has been possible through by using connectomics tools, by slicing the brain into thin slices and photographing each slice with the electron microscope and tracing all the wires. It's actually very, very laborious work. My group spent many years automating and developing the first pipeline for doing such reconstructions. But this work is not from my group. It's from the groups of Cardona and Samuel, who measured not just the existence of the connections between neurons, but also the weights of those connections. How did they know the weights of connections? Well, it turns out that in the fly, all the synaptic contacts are approximately of the same size. And size for synapses usually corresponds to their weight, W. And so by just counting the number of synapses, synaptic contacts in parallel between a neuron, that number that can go from 0 to hundreds, you could have an estimate of synaptic weights. So they just built a big table where for each connection, they had a number of contacts. And that is the estimate of a connection weight vector W. So that's how we got the weight vector W. Then we used another data set, which was produced by the Samuel lab. Sorry, I'm not advancing the slides.
Excuse me, I have a question.
Yeah, please go ahead.
You can measure, well, you said you can measure then the synaptic weights using the size of the synaptic, I don't know, the synapse. Yeah, but we're also talking about the neuron updating those weights. Does that mean that the neuron is making them larger or smaller, or I don't know?
So, so that's a good question. So the situation is somewhat different in flies and in mammalian systems. So in mammalian brains, the increase of the strength of a synapse in an experiment like long-term potentiation that I will talk about in a minute actually does make the synapse bigger. And it has been so reliable that people use the size of a synapse as a proxy of its strength in LTP experiments. So yes, they do. But in the fly, we found that the sizes of synapses don't vary that much. It's just the number of the contacts in parallel that changes. And so we can just use the number as a proxy of strength. Okay?
Great, thank you.
Okay, now I have some problem with my PowerPoint and doesn't advance. Let me.
So I have a question that probably resonates with Fernando's question, right? So in this framework, it seems like the weights are constantly changing and updating, right? So, but if we assume all the synaptic weights like are correlated with their size and their size doesn't change that much, right? So this like doesn't seem to work for this like optimization process, right? So is there anything I'm missing?
So there's two different scenarios that have taken place in mammalian brains and in flies. So in mammalian brains, you can just monitor the size of the synapse, and that will tell you its strength. And so the sizes do indeed change, okay? And that corresponds to a change in strength. In fly, the sizes don't change much, but the numbers of synaptic contacts can change. Like, for example, people have done experiments on fly visual system, and when they count the number of synapses at night, it's 20% lower than the number of synapses during the day. So the synapses come and go. Okay.
I see. Got it. Thanks.
Any other questions?
So when you talk about the size of the synapse, like, are you talking about, like, what do you mean by the size? The length, or what do you mean by that? Like, how do you measure the size of a synapse?
The easiest one is just to look at the volume of the postsynaptic density or the presynaptic terminal. And the most practical way, so you can do it in electron microscopy, but just outline the region where you see vesicles in the postsynaptic, in the presynaptic terminal, or you see postsynaptic density in the postsynaptic side. With light microscopy, you can do that by just labeling some kind of synaptic protein, like PSD95, for example, and then looking at the brightness of the puncta, which is proportional to the volume. Does that answer your question?
Yes, thank you.
Okay, very good. Okay, I restarted my PowerPoint.
It's okay. It's time to take a break.
Yeah, I think
we actually have a 10 minutes break in between.
Yeah,
so,
I was just about to say we should take a break now. Conceptually, it was just during a flow, but I think since we also have that, we can take a break.
Okay.
All right, so we can take I guess we can take a 8-minute break and we'll restart at 9.
Very good.